Prokaryotes are cells that do not have a nucleus and they do not have so-called membrane-bound cell organelles. And membrane-bound organelles are structures inside the cell with a membrane. They do not have that. But they have other interesting parts and components, and we're now going to label the prokaryotic cell. I will also tell you something about the function of these structures, but I'm not going to write this down separately. So if you would like to note down the function as well, you'll have to stop the video a little bit. So let's start off, and here in green, this is known as the cell membrane, and that is the only membrane that we have in prokaryotes. Inside here, generally, we do not find any membrane-bound organelles. Yes, there are some prokaryotes with exceptions. We're not going to talk about that. So the cell membrane, what does the cell membrane do? It controls what goes into the cell and what goes out of the cell. The flagellum, some prokaryotes have one flagellum, other prokaryotes have multiple of them, several of them. They spin very rapidly and they push the cell forward. So the flagellum is there for movement of the cell. Again, not all bacteria have that. Other bacteria move not actively on their own, but are simply carried around by the water or the wind or whatever. The cell wall in yellow over here makes sure that the cell does not burst, because the contents of the prokaryote are under pressure. The cytoplasm in here is under pressure, and the cell membrane alone would be too weak to hold it together. So you need a strong cell wall. Plant cells also have a cell wall, but the material is very different, so you cannot compare it. Even though it's called cell wall in plant cells and cell walls in prokaryotes, they're really, from the material that they're made of, not related at all. In here, these bubbles over here are actually not bubbles, but they should be also much smaller. These are called ribosomes. And prokaryotes, just like eukaryotes have ribosomes, prokaryotes, however, have so-called 70S ribosomes. They are a little bit smaller than the 80S ribosomes of the eukaryote. And the ribosomes are there for protein synthesis, so the cell uses the ribosomes to make new proteins. Inside here, the red part over here is referred to as the nucleoid. It's not a nucleus, it's a nucleoid, and the nucleoid is the region inside a prokaryotic cell where you can find its naked DNA. And the term naked means that the DNA is not wrapped around so-called histone proteins, which happens to be the case in eukaryotes. So eukaryotes do not have a naked DNA. So the nakedness does not refer to the presence of a nucleus or not, but it refers rather to whether it's wrapped around proteins or not. Here it's not wrapped around proteins. A plasmid, you see that it looks similar to the DNA, and the reason is a plasmid is a DNA. So plasmids are small pieces of DNA, circular DNA, floating around in the cytoplasm. The capsule, relatively thick over here, on the very outside, protects the cell, for example, from being eaten by the cell of the human immune system. Again, the thing is, not every prokaryote has a capsule, but if a capsule is present, then some prokaryotes use this to hide from the immune system. Pili are pili, singular pilus. These are protein structures which are used to attach the cell to a surface or to other cells. And I think I'm now going to make a drawing to simply illustrate this. This is some kind of a surface. Okay, some kind of a solid surface, and this over here is the prokaryotic cell. Then it has these spikes, these pili sticking out to all directions, and these pili can also connect the cell to the surface to make sure that it's not swept away or flushed away. So let's move on. Cytoplasm, of course. The cytoplasm is a complex mixture of many different chemicals, and many chemical reactions occur in the cytoplasm. The cytoplasm is made mostly of water, but it is not pure water. It's a very complex mixture of different substances. And here we are already at the next foil, which I'm not going to explain right now. I'm going to go back and I'm going to make a two-dimensional drawing of a prokaryotic cell. Let me find some space. I think I'm going to erase everything over here. Let's remove also some of the text. Okay, and I'm just going to draw it here because you have to be able to draw the prokaryotic cell, and you might want to draw it like this. Over here you have the cell membrane. I'm not going to label this now, you do this yourself please. You have surrounding the cell membrane, you have the cell wall, which of course is slightly thicker than the cell membrane, so that's the cell wall. And surrounding the cell wall, some bacteria, prokaryotes after all are bacteria, have a capsule. Okay, I'm running out of space again, does not matter. Inside here we have the cytoplasm. Inside the cytoplasm we have the naked DNA. Actually, it's a loop. It's a closed loop. We have some plasmids floating around. We have ribosomes in here. These are my ribosomes. You have extending from the cell membrane inside here these spikes, and we don't call them spikes. We call them pili. And maybe also a long or even multiple long flagella. And basically this would be a two-dimensional drawing of this three-dimensional drawing over here. And of course, every time when you draw something, you have to, of course, also label it. Now, looking at this drawing, I think that the capsule over here and the cell wall are drawn a little bit too thick. So you might want to correct this in your own drawing. Okay, so this was now a basic overview over the structure of a prokaryotic cell. And I'm going to show you everything again. And never forget to clearly label the drawings, especially the thin membrane can be easily confused with a cell wall. So make sure that if you label very thin or small structures, that the arrow really points at the object that you want to label. Okay, this was it.